the biological age and the chronological age are not the same. And understanding that gave me this view that if that's so, there's something that we can do about it.
When did your investigation into supercentenarians come into play?
We published that centenarians don't only live longer, they live 20, 30 years healthier. But I think the prospect of having not only your biological age, but also what organ might lead your aging, and can we focus on that, is going to be just terrific. Watch it for the next 18 months or 2 years. We'll have a major advance of how to measure aging that is clinically relevant.
I want to personally invite you to the Functional Medicine is Longevity Medicine Masterclass, a free online event featuring the leading minds in functional and longevity medicine. We'll explore why functional medicine remains the most science-backed path to extending health span. In a world chasing the latest longevity hacks, we're spotlighting what we've known all along. Healthy longevity starts with root cause resolution, systems biology, and personalized care. Join us live September 17th through the 19th at functionalmedicineslongevitymedicine.com. Thanks to our Diamond Level sponsors. Precision Analytical, creator of the Dutch test, is offering evidence-based training on key topics like hormone metabolism, interpreting Dutch test results, and therapeutic strategies, including hormone replacement therapy, all aimed at improving patient outcomes. Become a registered provider to gain access to an expansive education hub with ongoing content, including webinars and case studies. Visit dutchtest.com/courses to get started and grow your practice. For over 40 years, the foundation of Biotics research has been innovation and quality. Their goals remain unchanged: innovative ideas, carefully researched concepts, and product development with advanced analytical and manufacturing techniques. Biotics nutritional products are of superior quality and effectiveness. They are available exclusively to the healthcare provider. Find them at bioticsresearch.com. Thanks to our gold level sponsors. Vibrant Wellness, transformative lab tests for wellness. Find them at vibrant-wellness.com. Timeline, fueling cellular energy for a lifetime of healthy aging. Find them at timeline.com. Welcome to New Frontiers in Functional Medicine, where we are interviewing the best minds in functional medicine. And of course, today is no exception. I am here with Dr. Nir Barzilai. He is a true pioneer in the field of aging research. He's published over 330 peer-reviewed papers and is known for his groundbreaking work in extending health span and lifespan, both in humans and animals. He's leading the TAME study that's targeting aging with Metformin, which aims to prove that a single drug can fight multiple age-related diseases. Dr. Barzilai, it is such an honor to have you today on New Frontiers in Functional Medicine. I've been paying attention to your work for so many years, and gosh, I'm just really excited to dive in and kind of pick your brain in all of the area, well, as many of the areas as we can get to that you've been investigating. So welcome.
Nice being with you. Looking forward going to all the areas you want.
Yes, I mean you have you got famous with studying the supercentenarians, and I want to talk about that. But I know you've done a deep drill down into biomarkers, and and then we're going to translate and think a little bit about how clinicians can interpret and use your science today. And of course, we'll talk about the TAME trial and so on and so forth. So much to talk about. But first, I want to ask you about your journey into this field. I mean, you're really kind of one of the first biogerontologists. Like, you really played a pivotal role in creating what is, now extremely popular and getting a lot of attention, investigating aging.
Look, the journey really started much before that, because when I was 13, my friends were looking at their grandparents and saying, what happened to them? Where are they coming from? And for me, it was clear, not where they're coming from, but where I am going to, okay? And I think that was different. So it was always for me the biggest question in biology. You know, when I went to medical school and I learned that you can measure glucose or cholesterol or blood pressure, you know, you could put 100 people in front of me and I wouldn't know who has cholesterol or glucose, but I would know who's young and who's old, right?
Yeah.
When I went to my residency, I had a attending who, whenever I presented a patient, and by the way, I'm doing that thing in clinic too. This is a 76-year-old woman. He would stop me and said, "Does she look younger or older than her age?" And I thought, why does it make difference? But it does, because you can be 74 and all your systems are completely fucked up and there's nothing that you want to do. And you can be 95 years old and a little antibiotics will get you out of the hospital without hospitalization, right? That the biological age and the chronological age are not the same. And understanding that gave me this view that if that's so, there is something that we can do about it. If we can harness, if we can understand this biology, that means this biology can be targeted.
That's
And
amazing.
fast forward, yes, that's what we can do now.
But so you had that original insight that your destiny was to be like your granddad. And I know I've listened to other interviews with you where I think he was obese, if I'm not mistaken, and maybe he was a diabetic. So you at 13 had that insight. And then in residency, you had this extraordinary attending physician who asked you that question, do they look old or not? And that was the epiphany and these things shaped where you are. Okay, so keep going. Keep going with, so what next? Then what did you do?
So I wanted to study aging, and then, okay, how do I get there? Well, first of all, internal medicine, of course, and then which subspecialty, because I wanted to go into science. And I decided on endocrinology because I thought that I know a lot about those hormones that are declining with age, and maybe it's going to be just so simple as giving back those hormones and will solve aging, which happen to be much more complex and generally not the true. But then, you know, I went not only to endocrinology, I really went to metabolism, and I think that gave me the knowledge and the tools to start both science in animal models, in cells, and to think of how can we go from cells to cities, right, and to the world.
Yeah. Then when did your investigation into supercentenarians and centenarians come into play?
So, right around the time that I started with animals, actually, um, there is a famous experiment by Cynthia Kenyon, who was looking at nematodes, and she changed one gene in this nematode that caused them to live up to 5 times longer. Okay, one gene and longevity. And that was just so exciting. The idea that there's a genetic way, and it might be not that complicated to live much longer. And while a lot of my colleagues were looking, when it came to human, looking at diseases like progeria, you know, an early aging, and understanding them, I thought that's not enough. I knew that you can do something really dramatic in animals and screw them up so much that you'll get a phenotype. But for me, it was okay, I want to look at the other side. Okay, what causes longevity? And I thought maybe if I look at 100 years old, they could give me a hint if indeed they live healthy and not only longer.
Yeah. What was the gene she was looking at? What did she identify that allowed nematodes to live so much longer?
Um, it's called DAF2, and it's something that is not a complete knockout, but it decreases the insulin IGF-1 signaling pathway. You know, the insulin and growth hormone signaling pathway, which is just one in nematode. So it gets more complicated, but that's the pathway that it targeted. And there's actually, so the paradox for me coming from metabolism, that this nematode was actually insulin resistant, right? And we're all talking about insulin resistant and how it's a marker and maybe cause for lots of the aging, which is not, by the way, but anyhow. And not only that, the nematodes accumulated visceral fat, okay? They had fats around their intestine that became obese. And this paradox was even more surprising in the sense that it's against everything we knew. We knew that longevity is associated with thinness, with insulin sensitivity, and those were the opposite, but still they lived 5 times longer.
That's amazing because they had this gene basically inhibited.
Right.
Huh. And then so from that you yeah, I'm sure it was a while and many steps and many things happened, but eventually you began to look and you wanted to investigate the genetics of humans.
Well, what really happened, in my mind intellectually is to say, okay, there's the insulin signaling pathway and the growth, the IGF-1 pathway, and maybe we have to look at it differently. And what helped me think about it is the fact that in nature, the small, the dwarf live longer, the little dogs live longer, and the ponies live longer, and and so on. When you knock out, even in the lab, growth hormone or IGF, and when you don't have enough growth hormone action, they live longer, and when you have more, they live shorter. And so it was easier now to focus on the growth hormone IGF pathway rather than the insulin signaling pathway.
Mm-hmm. So talk to me about the supercentenarian work then, and and connect the dots with this original kind of epiphany.
So look, the most important thing, as I noted before, is, which we couldn't know in the beginning, is, do they live longer, or do they live longer and healthier?
Yes.
And and the answer is, and we did several studies and harmonized several studies, and we published that centenarians don't only live longer, they live 20, 30 years healthier, more healthier. Our control, by the way, when my centenarians were born, life expectancy was 50 years old. So they anyhow doubled their lifespan. But if you compare it to the people who were living right there at that time, and the Enhance study, by the way, which was at the same time that our Enhance 1 was at the same time, then they live healthier. In fact, after the age of 100, 30% of our centenarians don't have any disease. And by the way, I'm not saying that's true for every centenarian around the world, but that's my group. In other words, there is a group that can live healthier and can live longer. But that wasn't really the interesting thing. The interesting thing is that they had what we call a compression of morbidity. They were sick very little at the end of their lives, right? Those 30% that didn't have any disease would not wake up one morning. And this became the vision that we can live healthier, longer, and actually decrease our morbidity. The CDC, may it rest in peace, or may it resurrect, I guess. The CDC followed the medical cost in the last 2 years of life of somebody who dies at age 100, and it was third the cost of those who die when they're 70 years old. So it's not only my study, it's national study. And we could start calculating what we call longevity dividend. And there's this guy, Fitzgerald, and he said, guys, you don't understand what you're saying. You're saying, okay, those people are not in the hospital, but where are they? Because there's an economical value to the fact that they are traveling, they are shopping, they are building houses to the kids. So when you calculate that, the benefits for the economy are such that they're the solution. What you're doing is the solution to our economy, not the burden to our economy, which many people think.
Wow, that's pretty extraordinary. And when you investigated them, when you looked into the genetics, what are the key players that jumped out?
So I'm not going to let you out that quickly. I'll tell you
Okay.
a few other things before I
Okay.
answer.
Okay.
Because I have to convince you, okay, that
Yeah.
it's genetic. So we had three hypotheses. One is that they just did then what we know is right to do now, okay? They ate the right thing, they exercised, all the things we have to know. And I'll tell you immediately, the answer was no. 60% of the male centenarians and 30% of the women centenarians were smoking. Almost half of them were and are obese or overweight or obese, exercising even moderately, less than 50%, vegetarian like 2%. So the answer is, not that individuals couldn't have done it, but that wasn't the group. And again, it was like the enhanced one data pretty much. They were a little bit worse maybe than enhanced one. So it wasn't that.
I mean, that's fascinating. And I know that to be, I've heard you talking about meeting one of your first centenarians, and I think she was smoking or something.
Yeah, yeah.
And she
And she
said,
was smoking for 95 years. She was
yeah,
smoking.
that's amazing. Yeah.
Yeah, yeah. In fact, I met her when she was 100 years old, right? And in her apartment, and she was smoking. And I said, "Nobody told you to stop smoking?" And she said, "All four doctors that told me to stop smoking, they died," right? But then I started sending her reporters, right? When they interview me on my studies, I said, "Just go and talk with people, and you'll see what I mean." And she discovered that so by the way, after I visited, she said, "Okay, I'll stop smoking." Okay, but then when reporters came, she understood that she's a celebrity for being 100-year-old and smoking. And by the way, she lived to be 110 and she resumed smoking. Okay, so I always said, I don't know when you'll die, but without the cigarettes, I bet it would have been much longer. I'm not sure. I think the point of this is in fact that for those people it didn't matter. And you could see from time to time in the papers about centenarians and what they think, right, is the secret for longevity, and it makes no sense.
Yes.
I never moved, I just stayed like that, or I ate cakes all my life, or I'm drunk. Okay,
Yeah.
it really,
Yeah.
for them it didn't matter, which is not the same as for us it matters.
Yes.
So the second thing that we looked at is maybe, we know, and we use genetics in clinics now, we know that there are lots of SNPs that are risk for diseases, for cardiovascular disease and for cancers and for everything. And I'll give you just example that happened in after the 44 first centenarians that we had. And it took us great effort and it cost fortune to do a whole genome sequencing for those 44 centenarians.
Right.
And without control, okay, only the centenarians. And the only thing we could ask is, do they have any of those SNPs? Maybe they have the perfect genome, okay? Maybe they just have one of 10,000, just don't have any of this stuff. And that's why they're healthy. We didn't expect that 44 centenarians will have over 260 SNPs, that each one of them was supposed to make them sick. And they're 100 years old and they're not sick. And that includes, ApoE4, for example. Okay, ApoE4, you're supposed to be demented at 60, 70, be dead at 80. So they're 100 years old and not demented.
They're
Okay?
smoking, they're clear as a bell, and
So,
they're ApoE4 positive.
right. So those two things just to show you that they must have something that slows their aging. Okay.
Yes.
And the first thing that we picked. So here is another challenge.
Yeah.
If you take a blood sample from centenarians, and something is high, okay, what does it mean? On one hand, this centenarian statistically had a 30% chance of dying the next year. So maybe it predicts its mortality. Or maybe it's what got them there. How would you know? And the answer is, when I initiated the study, I wanted their offspring, okay, that inherited, half, let's say half of their longevity. And those who are married to their offspring, who share the environment, in case their they don't have longevity themselves. Okay, we would study them, but not at the same group. And one of the things was that for the phenotype, it will be if the offspring that are 20, 30 years younger have this phenotype, it suggests that it's important. Okay, if they don't, then I don't know what it means to be a centenarian and have something basically at the end of your life. And the one thing that came up almost immediately, I think after like 50, we had 150 people in our studies approximately. The HDL cholesterol of centenarian was average, between 45 and 55 if you're male or female. But their offspring had twice the HDL. I mean, we had offspring with 140 HDL. Now, HDL goes down with age by 5 points every 8 years. So we hypothesize, but cross-sectionally, it's the same. How can something cross-sectional be the same but change with age? Maybe because if HDL goes down, you die, and the average who are alive are keeping the HDL normal, right? That there's a longevity effect of HDL. And that supported that because the centenarians now had normal HDL, but their children had high HDL level. And that led us to the first two genes that we discovered, functional genotypes that we discovered that are also now being made into drugs, not for longevity, but might be for longevity.
Interesting. So you would say, and also HDL particle size, correct?
Correct.
So quantity and the sort of the type of HDL.
Correct.
They just have a ton of it.
Right, and they had big particles.
Wow. And that continues to bear out, like that original hypothesis that when did you document that? Like when did you publish on that particular gene?
I think in the early 2000s.
Wow. And that's and you would still stand by that data as a key player.
Oh, yeah. It was validated. Not only that, the genotype that we discovered was validated by some, but not all. Okay, but was validated genetically, and as I said, two drug companies looked at my data and decided to do something about it. One is a CTP inhibitor by Merck, and one is a ApoC3 inhibitor by Ionis. They were ISIS then, but now they're Ionis.
Fascinating. Okay, and the CETP allows for the higher quantity of and larger particle size of HDL, and the ApoC3 drops. Well, keeps triglycerides low.
Yeah,
What's the mechanism of that?
HDL and triglycerides. Both of them have phenotype of HDL and triglycerides.
Interesting. That's fascinating. So now, can we translate that immediately into action for the mere mortals? I mean, how would you translate that?
So the drugs that were developed for cardiovascular protection. Okay. One of the thing that we noticed, the people that had this genotype for CETP had much better cognitive function. And we told Merck, as they discovered that, we said, why don't you try to design a study and look at cognition? Okay? And they looked at cognition, but the study was people who were 50 years old. And so they don't do much with cognition in a year's study or so. Okay? Yeah. So they missed it. But now there's actually articles suggesting that taking the drug is protective from cognitive function. We have to see how it rolls, but for us it was really an aging drug, an aging, not even those with the genotype also had less cancers than others. So at least in our hand, it looked like it protects from age-related diseases rather than from just one important
just keeping your HDL quantity up. Right. It has a broad spectrum effect.
Right. And by the way, as you noted, I want to double down on that. The HDL was a phenotype, whether this is the important thing or whether it's the large HDL particle, or maybe it's the large LDL particle. I mean, there are so many things associated that you couldn't say what's most important. And also we know that this is an inhibitor, but if you inhibit this pathway is a way to get rid of lipids. So it has to be some modulation, and not a total thing. But you ask practically, those drugs are going to be marketed soon. Phase 3 is years ago, but they had to find the right combination and the right time to bring the pharmaceuticals are doing that. But it looks like it's coming around, and it will gain more indication, and keep an eye on it.
Yeah, absolutely. Very interesting. How are these second generation of your original centenarian cohort? Like, how are these folks who,
So
yeah,
they're much, so they have half the cardiovascular diseases. They have half the cognitive decline, roughly half the cognitive decline, half the mortality. Um, I was just in a meeting where they presented data from Europe on centenarian. It was a centenarian meeting, and they basically showed that if you have two parents who are over 100 years old, it will contribute 24% to your longevity. So let's say, compared to others, let's say if in your environment people live to be 80, then you're likely to be, 80. So it's 16 and 24%. So it's about 100. You're going to be about 100 years old. If only one of your parents, it's 13%. Okay, so it's still into the 90s. But interesting, even the third generation has effect of longevity of, like 8%. So the inheritance of long life is really very impressive, no matter when the other parent died.
it's pretty interesting. That's interesting. There's a lot of effort, of course, because many of us don't have, supercentenarians or centenarians in our family. So there's a lot of energy around, your genes not being your destination. And, there's much intervention that we can do, of which, of course, is true. But it is interesting to see. But what about, the caveat is that these are healthy centenarians, correct? I mean, I have friends who've got long-lived parents, but, one in particular, a good friend of mine, his mom spent, the last 3 years in bed. And I think she passed at 103, but
Right. So she wouldn't be in my study. Right. And yeah, let's make clear. I didn't present all the centenarians in the world, right? I presented a selected people who actually had to be consider themselves healthy and independent living
yeah,
at age 95. After that, it didn't matter. Okay. But that they had clearly long health span. So I'm not saying all the centenarians are like that, but we were going to pick the best of the
sure.
best in order to find also the best
No,
findings. So I'm not, so people are saying, no, I know another centenarians, and I totally agree. I would just argue one thing.
hmm
Even if this 103 has spent the last 3 years of her life, in bed or with lots of needs, um, still the percent of health span versus lifespan is quite incredible, right?
yeah,
Um, and it worries me. I think we don't have a good exit strategy. Okay, and I think that's a problem, and I think we have to fix it. I think Kahneman, I don't know if you know this Nobel laureate for economy, who at age 90 basically went to Switzerland to have an assisted suicide.
interesting.
And he wasn't like that. He wasn't demented, but he said,
Sure.
"Look, I'm 90 years old. I have a renal failure. So
hmm
in the next year, I'm going to dialysis, and I know what it means, and I know there are sepsis and stuff. So I see the future. I'm very content with my life. I'm going to separate with everyone, and I'm out of here."
Yeah.
Um, And I really want to work on extending health span,
Yes.
and with the knowledge that the morbidity will decrease, but not always. And so we need to think of an exit strategy.
That's very interesting. That's very, very compelling. Yes, I think it's extraordinary that you're bringing that conversation front and center because a lot of us, I think, just secretly hope for a compression of, morbidity. You hope for it. But to your point, I mean, how do you, I mean, how do you control for that? We just don't know. And yeah, really fascinating, and I think that's fair to consider. So other genes I want to talk to you. You looked at IGF-1 receptor in this population as well.
So it's not so. I'll make it more dramatic even.
Hmm.
60% of the genetic features, 60% of centenarians have something genomic that inhibits the growth hormone IGF signaling pathway. Okay, it's the most common abnormality in our centenarians. And even women with the lowest IGF-1 level, with the lower half IGF-1 level, so they're already 100 years old, they live twice as long as those with the upper IGF-1 level.
Fascinating.
And those women have better cognitive function, and they don't pay on muscle. They have the same physical activity, so it's not that there is any exchange. And in order to understand what happens really, we actually went to the UK Biobank. The UK Biobank have, I don't know how many by now, millions of people with basically their electronic medical records with a lot of things that they're doing extra. And like 250,000 of them have IGF-1 level, which is a good indicator of the growth hormone status.
Yeah.
And we basically showed that when you're young, high IGF-1 level is protective for everything and for mortality. By the way, not for cancer, but all the other diseases. You have high IGF-1 level, you're doing better and you die less. And somewhere at the age of 50, it flips. So the same IGF-1 level when you're over 50 is associated with all diseases and mortality, except cancer, because high IGF-1 is bad when you're young and when you're old. But this was kind of the striking finding, and it kind of intuitively explains that when we go through this threshold somewhere between 50, 60, it's individual, and we start to break down, then it really makes no sense to invest in growth. We need the energy to be invested in stopping the breakdown. And maybe this energetic shift really explains why it's so important to have lower growth hormone access when you're older.
Explain that. I mean, how do we think about that in terms of? I mean, I know Walter Longo, who's been on my podcast a couple of times, of course, talks about lower growth hormone being protective, and his focus is in cancer. There's a lot of energy towards maintaining growth hormone levels as an anti-aging strategy, and clinically, using growth hormone therapeutically, using peptides that will increase growth hormone, really consuming high quantities of protein to maintain muscle mass, which will, indirectly likely influence growth hormone if you're laying down muscle. So there's that. And then also, I mean, of course, you're familiar with Greg Fay's study and, Steve Horvath, and using growth hormone injections in an older population and regenerating the thymus gland to a certain extent, and reversing the epigenetic clocks that that they measured. So, I mean, how do you reconcile what appears at a glance to be kind of conflicting?
So yeah, and it worries me. So let me explain a few things. First of all, I have to say that the effects that we were showing were were larger in females than males, in part because we have, for every 100 centenarians, there's 80 women and 20 men. So
Yeah.
we don't have enough power. But it looks that it's more female specific. In fact, we took it to the lab. What I mean, we did now a reverse translation. We found these findings in centenarians. We noticed that there was a development of IGF receptor inhibitors by, we went to Amgen, but other companies that were trying to use it for cancer, because cancer express IGF, so they tried IGF receptors, so they tried to look at it, and by the way, it didn't work. But we had this IGF receptor antibody, we murinated them, and we injected it to old animals. And the female animals lived 10% longer, but much more impressive was their health span. They were running better and cognitively better, and everything was better there. It didn't work like that in male. So I'm saying there could be a sex-specific effect, but that's not to say that more growth hormone is necessarily better. The experiment with the thymus, it was interesting. I could simply say metformin took away the actions. Metformin was the needed one.
Right.
But there are a few issues. Let me just say the first one. We know that the epigenetic wasn't the right thing to do, because the epigenetic test was done on white blood cells, and there was a very different change in the composition of new cells that were created. So it really, this clock didn't really reflect true aging. It reflects the biology of new cells because of the thymus. And by the way, I think it's a great target to do thymus and to see what happens.
Yeah.
The second thing is that the fact that usually it's good to have a low IGF-1 doesn't mean that there's no indication from time to time to use growth hormone. Growth hormone was used after stroke with some success, okay, for a short period of time. Mm-hmm. So I think there's a chronic basis of what you want your IGF-1 level to do. And there are cases where you can try a growth hormone. And there's also the sex differences that I'm not sure. By the way, there are conflicting results on that. I mean, I had a really cool study on males, okay? Only males. We noticed that 12% of our centenarians have deletion of exon 3 of the growth hormone receptor. Okay, wow. And by the way, we replicated it in several studies, several other populations, all the same results. It was 3 times. This is not a very rare deletion, but it's like in 3% of the population. So it was highly relevant in our centenarians. All the longest living in 4 studies had much more of that. So it looked like it's important. And when we measured the IGF-1 level in those people, it was really low, significantly lower. So it made sense. It took us 10 years to publish this paper because our centenarians, though they had lower IGF-1 level, they were 2 inches or more taller than anyone else. And it made no sense to us. Like, what am I going to say? What blah, blah can I say? Until we actually took the cells to the lab, and we showed that the cells from the centenarians, we have immortalized lymphoblasts. The cells, when you incubate them, they secrete their activation of the growth hormone receptor is decreased. Okay, so it's a problem. But when you incubate it with growth hormone, it was amazing. It was flipped. In other words, the activation was doubled, and so was the proliferation.
Interesting.
So I don't want to confuse you so much, but this is what happened. When they went through puberty and growth hormone was high, the receptor was hyperactivated. And when growth hormone was low, the receptor was hypoactivated.
Right.
So they grow taller, but then throughout their life, they had lower IGF-1. Again, it's men. So in this example, it's men. So I'm really not comfortable to really talk about the sex difference totally. It might be more confusing or needs more study. Hey, that's why we called it research. If we found it the first time, it will be only search, right? So
Right.
I'm not sure about it, but I would just state that basically chronic growth hormone therapy is probably dangerous for everyone.
Is there a gap in your health and longevity strategy? MitoQ mitoquinol is an advanced antioxidant that targets the inner mitochondrial membrane, reducing reactive oxygen species by 48%. This patented compound is backed by 30 years of research and 25 clinical trials by Harvard, Yale, and Oxford. MitoQ supports mitochondrial and cardiovascular health, metabolism, and DNA stability. Learn more at mitoq.com/Dr. Kara. That's mitoq.com/Dr. Kara.
You've likely heard about the longevity benefits of peptides. They're in skincare, supplements, they're everywhere. But not all peptides are created equal. Developed by skin longevity scientists, OS1, which is found in OneSkin topical supplements, penetrates deeper layers of the skin, impacting cellular level biomarkers for visible improvements in lines, wrinkles, and thinning skin. Ready to experience OS1's benefits? Use code Dr. Kara for 15% off your first order at oneskin.co/DRKARA.
So I have to tell you this anecdote. It happened not long ago. I was talking to lay people in Florida, and I was talking about this example. And there's a guy. There were questions. There's this guy, old guy, and he gets up and he says, "Well, I'm 95 years old, and for the last 15 years, I'm injecting growth hormone." The last question you want to deal with, of course, to him, I said, but I already told you that centenarian doesn't matter what they're doing. Okay, if they have genes to be centenarian, they'll get there. Okay, that was how I came out of it. So after that, he comes to me, and behind him there is another old man, and he said, "This guy is my doctor." He's the one who's mentioning me growth hormone. I said, "How old are you?" He said, 88. "Are you taking growth hormone?" "Yes." "Your patients are getting all growth hormone?" "Yeah, I'm giving all my patients growth hormone." "How many of them died?" "Nobody died." He said. So, people have their own experience, and I've no doubt that there's some. Look, the nice thing with growth hormone, the reason that people believe in it is because they're losing subcutaneous fat, so they're, and they're around their muscles, so they actually look better, okay, because it's lipolytic, okay. But I think for many other purposes, it's not really safe to give growth hormone, and unless there are many studies like that, unless there's another study that shows that there's some population or some conditions, I think this is a very fundamental problem in aging, and I think that low growth hormone is really very important for my centenarians to get there.
Yeah. Okay. All right. I hear the point taken. So there's a time when we want it, we want robust amounts, and our body responds and it's ready. And there's a time when we want to put our energy towards preservation and not growing. Got it. Um,
By the way, you see, you got me to be a scientist now, and I'm kind of regretting making it too complicated. You'll decide.
no, no, no. I think it's. I think it's really interesting. It's very. I mean, what you're saying is. I mean, well, as my mentor always said, embrace the uncertainty. Like it's not a straight line. It's not a straight line answer. It's really, but I think there is a fundamental take home around, around there being a time and a place for growth hormone. I do though, epigenetic clocks notwithstanding, I do think it's interesting though that Faye that Faye and his team have regenerated thymus function, and it'll be interesting to
Yeah,
watch that.
I think that's a cool idea,
Yeah.
and is worth and was worth studying.
Yeah, that's right. And it's and it's short term too. It's not a chronic use of growth hormone. All right, so let's go to um well, actually, I want to just button up a little bit on the CETP gene. So this healthy cholesterol pattern, how would you translate? What would be your action for your regular patients? How do we advise people? I mean, the usual things of exercise and healthy dietary pattern and using medication if indicated to maintain healthy cholesterol levels, et cetera. Like these people with fabulous HDL and fabulous LDL, would you use the standard tools that we're using, lifestyle and sometimes medication?
Look, I think what's common to all of us, the longevity doctors and the functional doctors, is that we believe that exercise, diet,
Yeah.
sleep, and social connectivity are important for health, and that you can not only that, but you can optimize that. By the way, I used to say maximize it, like do more, but it's really not true. It's optimized. You don't want a 90-year-old man to run the marathon eventually, right? That's not a good optimization of his health. Um, so we're talking, okay, and this thing you optimize at any age, right? Now you cross the age of 60, and and you feel something, I've been there. um, you cross the 50s, you start seeing what you're starting to lose. You're trying to maximize everything, and in my mind, that's the point where we'll start to do prevention. And prevention, I mean with medicine, and I mean before you get disease. If our
Hmm.
whole hypothesis is that aging drives diseases,
Hmm.
then you need to interfere before diseases, just like you do with cholesterol,
Yeah.
okay? Before you get the heart attack and not after. And we're trying to figure out who, when, and which drugs. But there are 4 drugs now that for us have passed the test of gerotherapeutic, that they're actually targeting all the hallmarks of aging. When you give it to animals, the animals live longer and better. And when you look at human studies, at clinical studies, you give this drug for one indication, and hey, in clinical studies it prevents variety of other diseases. You give it for one indication, and hey, in clinical studies it decreased not disease specific mortality but overall mortality. So there are 4 drugs that are out of scale of 12 are 11, 12. And I
Okay.
think the challenge is, okay, if you need this treatment, which drug should you get? And of course, we even don't know about combination, and we don't know about huge amount of drugs that are now in biotechs and are going to be, really more dramatically changing our health span.
We'll talk about these 4.
So it's metformin, SGLT2 inhibitors, GLP-1 agonist, and biphosphonates. Oh,
Interesting. Okay.
and we didn't, rapamycin is not getting there because there's no clinical studies on that. But I want to say, even before that, nutraceuticals are a real problem for me, and I think we should use drugs much before nutraceutical. And there are many things with nutraceutical. I mean, we don't know what's in the bottles even, and there's no clinical studies. And then there are combinations, and we don't know anything about the combinations, except that we know that there's a JAMA study about a year ago where they showed that they followed 12 million people, no, 7 million people over 12 years, and they showed that those that had multi-supplement had 4% increase in mortality. This is so surprising because those people usually also exercise and diet, and still you had the signal. So I would say nutraceutical need to go through clinical studies, and we're kind of trying to convince the FDA to have a supplement that were studied part, because because that will be important to give to our patients. But even before you're considering supplement, you should consider FDA approved drugs that are already, they're safe. There's a lot of data about them and combination and their side effects. And also you want doctors to lead this longevity medicine. You don't want to kill many people on the way to success. So I think it makes much more sense for me to consider FDA approved drugs than a combination of supplements that you don't know what they're doing.
I will push back a little bit on that. I hear you. I do. By the way, everybody, we'll corral together all of these papers that we're chatting about and make them available to you on the show notes. I think that JAMA multivitamin study wasn't it using? I think it might have been using UK Biobank data as well, if I'm not mistaken. Do you happen to know that? But I'll find it.
I didn't think so, but
Okay,
I'm not sure.
that's fine. Yeah, okay. Well, we'll find it. I remember when it came out and having some questions around how much I
By the way, those are, obviously those studies are complicated, right? It's not a
yeah,
clinical study. So
that's yeah.
there always you can find something, but it was worrisome.
That's I respect your response to that, but you can't be throwing, say, vitamin D and omega-3 fatty acids in that camp, are you? Do they
Eh,
fall in your
yeah,
nutraceutical
look,
bucket?
vitamin D helps only people with osteoporosis. I don't think there's any study that shows, not on the biology of aging and not on clinical, that vitamin D is needed. I have, believe me, the lowest
Interesting.
vitamin D in the world, and
Do
I'm not
you?
taking it. I'm actually thinking that I think we don't understand enough about vitamin D. There's a feedback as an endocrinologist. I think there's a feedback mechanism that they're missing. I mean, why my vitamin D will be low when I'm suntan? I don't know if you notice it. I'm suntan. My PTH is good. My calcium is good. And my vitamin, I think it's because I don't need I don't need vitamin D. I understand the association between vitamin D and diseases, but that doesn't mean that vitamin D is solving the problem. I think we're missing maybe physiology, maybe other things. So I'll tell you that the other reason is that I have a coronary calcification, and I'm thinking, you know, maybe if my vitamin D is there on purpose, why am I going to increase calcification? I mean, it's a little bit personalized medicine for me, but and my doctor wanted to give me vitamin D, and I said, show me. And he said, well, osteoporosis. So I did a bone scan, and that's another thing. My bones are 2 standard deviation thicker. Okay, so I have some calcium problem. I don't know that I need vitamin D.
Ah, well, maybe vitamin K. If you came to my practice, I would probably want to give you both.
Oh, okay.
K2, K2.
Okay.
But we can have that. We could have that conversation later. Fascinating. Well, geez, I don't know. Maybe we'll have to have. Sometimes we do podcasts after the podcast. I'll pull together my vitamin D research and talk to you. It'll be interesting to hear how people respond to that. It's such a that's very interesting coming
I
into
know.
the belly of the
I'm
beast.
on the lion's den. I know that,
You
but
are.
but it's more. I really wanted to say, even if you have indication and even if you have there, why don't you consider FDA approved drugs?
Oh, fair. That's fair. No, no, I think that yeah. No, I don't think anybody in functional medicine is gonna kick, a well-prescribed drug out of a program. No, I agree with you. But let's talk about metformin because I mean, you have been working on the TAME trial. You've been looking at metformin for many, many, many years, and I would love to understand that. I want to understand, how did metformin get into your world? Were you influenced by the centenarian data? I mean, going back to the worms who were like insulin resistant and had which sounds so funny to me, visceral adiposity, truncal adiposity, and and yet they were living for a really long time. How did you end up getting into really aggressive glucose control? And your list of, top drugs are all about really aggressive glucose control. And of course, keeping your IGF, under control is going to play a huge role. Keeping your growth hormone low is going to play a huge role. So you got into this whole metformin direction, and I do want to hear about it. And I also want to throw out, as you're talking about it, at some point, I want to talk about using it in healthy individuals, and the issues with mitochondrial damage. So, boom,
Okay.
go to it.
Okay. So, so the serendipitous story is that so I was trained in Israel, and there that was in the 1980s, mid 1980s, and metformin, like everywhere in Europe, was available for diabetes and was clearly an effective first drug choice for metformin. It has been for so many years.
Yes.
I came for a fellowship at Yale in 1987 because I was interested in aging, and I wanted to look at insulin resistance and aging. And the project that was available, by the way, I was with a very famous guy, Ralph DeFronzo, he's still very active. And he said, the project for you is to find the mechanism of action of metformin in type 2 diabetes, okay?
Yeah.
And I'm saying it's funny because some people say you're wrong. The mechanism of action of metformin is this. And I said, yeah, who's the author of this study? And then they discovered it's me, right? So I can I can increase my knowledge, and I'm not taking back anything I wrote. But okay, so metformin became available in the United States only in 92 or 93. Okay, like years later, but the story of metformin is that it's an extract of the French lilac, and it was available but it's not nutraceutical. Okay, it was modified. There are actually several cousins that had several other side effects until you go to metformin. But what was it used for? It was used for osteoporosis, osteoarthritis, to prevent flu. They tried it to malaria. It was an anti-aging drug, okay? And then somebody noticed that people with type 2 diabetes, it lowers their glucose. So it was hijacked for glucose, and the diabetologists are pretending that it came from diabetes. But no, it didn't come to diabetes. But when it came to diabetes, people noticed that people on metformin are doing better. Cardiovascular disease in clinical studies, cardiovascular disease, cognitive decline, and more data has accumulated to the effect that metformin now is the first drug. So first of all, it was repurposed, right? It's for diabetes. FDA indication is only diabetes, but it's repurposed. So polycystic ovary syndrome is the first drug of choice. It was used for obesity, of course, not anymore, but it was for obesity. Clinical trial, the DPP, it was prevention of diabetes, so it's for pre-diabetic. Now, in New York, 2 summers ago, it was prescribed for COVID patients because there was the clinical studies that showed that metformin prevents hospitalization, death, and long COVID. Okay, if it's given in the first few days of death. Also showing you metformin is not only metabolic, okay? It's also infection. It's also inflammation. It's also other hallmarks of aging.
It's kind of fascinating. Like its original indication was osteoporosis.
Well, I didn't say indication. It was used that way, right?
Okay.
It was in Europe. There's no FDA. There's no indication, but that's how it was used. You'll see lots of studies on that. And then the studies were on diabetics. And by the way, now macular degeneration. A lot of patients with macular degeneration get that. So it's legal to repurpose FDA-approved drug. It's legal, and it's being used. There are more people without diabetes using metformin than with.
So you You can dance through the hallmarks and basically point to metformin, you would say?
All of them. Metformin is maybe the only drug that we know hits all the hallmarks. Okay, rapamycin misses one. GLP-1, when I say miss, maybe it hasn't been studied yet. Realize that metformin, is a 100-year-old drug, right? So there's a lot of studies, lots of people have looked at that. And you can look every month, there's a new study on metformin, and it's important. Okay.
Right.
So that's kind of the story to metformin. I just want to say the TAME study is not about showing what metformin is doing, because it has been done. The TAME study is about showing the FDA that we can target aging and prevent not one but several age-related disease and mortality. Okay, that was the only point. And also for other pharmaceuticals, like Lilly is considering to do a TAME study on GLP-1 to just show the way of how do we test a drug that targets aging. By the way, it's third of the price or quarter of the price of developing a drug for a single disease.
Yeah.
Because if you take population of 65-year-old and you're agnostic to the disease, you're saying, I'm giving you metformin and I don't know which disease you're going to get first. If your mother is diabetic and you're obese, you're going to get diabetes, but whatever is it that you're going to get first, we're going to prevent it or delay it, right? So that's the idea. It's not a very expensive study, except when you have to raise your money yourself. And it's a template for the industry, and it's a template that will be used widely, I presume, because the FDA has said, okay, bring it on. So
have you started it? Are you recruiting?
No. And we're waiting. So many things happened.
Yeah.
We had a billionaire that was going to fund this study, and he lost his billion.
Oh, jeez.
Then
Minor.
COVID happened. Then we had
Yeah.
another big foundation that was going to fund it, and then they pulled back. And now we have several other options, and I'm not going to tell you which they are because I don't want to be again quoted as next year.
Yeah. Yeah. But ultimately, you're going to be looking at a healthy older, like a multi-clinical trial. These are going to be healthy older, middle-aged people, I believe, right? And they're going to receive metformin.
We don't want to recruit the people who are going to be centenarians, right? So, in fact, it's more of a secondary prevention in the sense that they could have had a heart attack already, or they could walk slowly, or, something like that. It's not that there's no sign of aging. In fact, if you're totally just a 40-year-old, biologically, you're not going to get into this study. So you have to show something. But basically, it's a secondary prevention of aging. I mean, it's aging when the secondary prevention is age-related diseases.
Okay. So they have to have something, but you don't really care what. But they're not coming in with two parents who are 120.
I don't care. It's more that I'm agnostic, right?
You're agnostic.
It doesn't matter. This disease is going to come because you have aging going there.
You have aging.
And that's the risk factor.
Right. Okay. All right. Well, let me ask you. It's funny. I was thinking about berberine because berberine recently made JAMA. There was a review on it, which is extraordinary because, of course, we use a lot of berberine in our world. I'm sure it has an influence on blood sugar. But I would imagine that you're not the biggest fan of berberine, given your nutraceutical comments earlier.
No, look, I don't know what berberine is doing, but I don't think that it's metformin. Okay,
Yes,
I don't think it's metformin.
Right.
And even metformin,
agreed.
is kind of funny because although it's one drug, it hits the hallmarks in two different pathways. Okay, and metformin can work even in cells that doesn't have mitochondria. So I don't know if mitochondria is always necessary. Metformin can work also if you knock out AMP kinase, which is a major mechanism. So when people came to me and said, we're going to imitate metformin, we're going to target the mitochondria, I'm not sure that that's going to be metformin necessarily. Okay?
Can you
That,
give?
that's
Oh, go ahead. Finish.
I finished.
Well, okay. I mean, can you just like give me a rough of the metformin mechanism since you're the one
Oh,
who identified
I'd love
them?
to give you this slide that it'll take me 10 minutes to show how it goes. But the important part, for metformin, there is an important transporter that gets it into the cell. It's
Yeah.
called OCT1. I think it's important to note also because between 3 and 6% of patients with metformin have diarrhea that lasts over a week. And the reason is that there's some genetic mutation in OCT1, and sometimes there's no expression of OCT1 for whatever reason. And if you have mutation or no expression, your metformin is not going to be transferred from the intestine. And that's why you get diarrhea. So it's not going to help you anyhow. You don't have to say, oh, I'll have diarrhea, but I want metformin. It's not going to go in. Okay?
Okay.
Okay. So then it enters the cells and goes to the mitochondria. So let me tell you something really dramatic. It's a weak cyanide. Okay, metformin. It's a weak cyanide. And when I say it's a weak cyanide, think of those centenarians, 8 centenarians who might have a mutation that mimics metformin. What if I give them metformin? Wouldn't I make it a big cyanide? Wouldn't I kill them? I think if people don't feel good with this drug, they shouldn't take it or take low. But it goes to the mitochondria, but that's very important because in the mitochondria, two things happen. There's an energetic shift that increased AMP kinase and does all this pathway that is involved in improving insulin action, the anti-diabetic effect, but also inhibition of mTOR and do what rapamycin is doing, and by that targeting half of the hallmarks of aging. On the other side, because it kind of stunts the mitochondria, there's also less oxidative stress and less inflammation and less of other things that are related. It's not senolytic, but it's senomorphic. It decreases secretion of subs. So it has basically a lot of effects, probably through those major pathways, and maybe not all of them. People are saying, you're arguing about what the mechanism of action of metformin, but we're not arguing that they target all the hallmarks of aging, okay? And really, this is what's the important. I think some of what we're seeing is just secondary. We don't know how it starts, but that's how it ends, and that's kind of metformin. But saying that, an important study came from Charlotte Peterson, where she had a grant to show that she can exercise 75-year-old people, and her hypothesis, and half of them on metformin, and her hypothesis is metformin will have better results on everything because it'll be synergistic to exercise. Exercise and metformin will be great. What she published is a paper that showed that the people on metformin, their muscle grew less. Okay? Their muscle grew less. So for that, you have to go and read the paper and get to supplement 4. And supplement 4, it's interesting because she measured the strength of the muscle by several ways, and they were not different between the groups. So in my mind, if you have less muscle and the same force, then every gram of muscle is better. So I said, give me the muscle. And this paper that I've done with those samples is less known, was not covered as much. But we found that on one hand, it decreased mTOR genes that are important for the muscle to grow, to hypertroph. But on the other hand, it changes genes for autophagy, for inflammation, for many other things. So in fact, the quality of the muscle, so there was a trade-off. The muscle was maybe not as big, but it was better. Okay? But please understand, you know about the X prize?
Yes, yes, yes.
Okay, so the X prize is we give you a year to decrease your biological age by 20 years. Okay? Good, good luck. Good luck, but there'll be results. The X prize wants to have really three things, to concentrate on three things: immunity, cognition, and muscle. Please understand that they can be contradictory. If muscle is about making the muscle bigger, then it's not going to happen whenever you use metformin or rapamycin. But it's going to be more effective if you're going to look at immunity and on cognition, okay? And I think the lesson from it is that for longevity, we really have to do several things, okay? In other words, if you're on metformin, you better exercise your muscle, or on rapamycin, okay? Maybe there's a better time and stuff, you have to figure it out, but and that combination of drugs might help to make you stronger on one that is deficient. When I'm on the scientific advisory board of X Prize, and I was going from company to company to ask them, what's your weakness? Okay, because you're going to be graded on all of them. And if you have a weakness, why don't you team with another team that has another weakness that you don't? And maybe this combination is going to really take you somewhere. So another thing that I've done in my studies is I took, that's my studies, we took, and I'll describe, we did several studies with several gerotherapeutics, but I'll describe the metformin one. We took, I think, 15 people to a crossover, double-blind crossover study, where for 6 weeks they got metformin or placebo, 2 weeks, crossover, nothing,
Washout.
and then washout, and then those with placebo got metformin, and those with metformin got placebo. And at the end of each study, we took a biopsy from their adipose tissue and muscle, okay? And we were really interested if the biology of aging has changed, if the transcript if our transcript have changed. And the interesting thing is that the transcript has changed, of course, in the metabolic genes, but also in other genes. BRCA1 expression has changed. Lots of things that are not just the metabolic have changed. Again, making the argument that this is much wider effect than just metabolism.
So even as it's been kind of vilified as being a mitochondrial toxicant, an electron transport chain, toxicant, your argument is just strongly against that. And
there's two things that are actually very important to say. First of all, if you do VO2 max and you're in metformin, you're not going to get to the VO2 max, right? Because, now we don't need VO2 max, okay? VO2 max is a great provocative tool, but we don't need VO2 max to function. What happens below the VO2 max has a lot to do with aging, okay? And maybe protects you from harms of exercise. So, and the second thing that's really related, metformin is for old people. It's not for young people. Okay, metformin lowers IGF-1. Okay, so we don't want it because it's always better when you're young to have IGF-1. If you're exercising and want your muscle, you want to build muscle, don't take metformin. Okay, I think that's very important. I think, Peter Attia took metformin when he was young and really badmouthed it, and it's like, how silly can you be? Of course it's not good for you, okay? There's antagonistic pleiotrophy hypothesis of aging,
Yes.
right? Things that are good for you when you're young are against you when you're old.
Yes.
And metformin is Metformin is good for you when you're old. Look, if you're diabetic, if you're PCOS, yeah, there is indication. But
Yeah.
otherwise, it's a potent drug,
Yeah.
and you have to use it right.
Yeah. Awesome. Good. That's great. Um, okay. So, like, I guess we're just pretty much in wrap up here. I could continue to pick your brain on it, but I want to maybe just maybe just ask you a couple questions about the Biomarkers of Aging Consortium and, just any highlights to share from your work there. I know you, I guess, really before Schneider, you were identifying those sort of aging spurts, looking at very omics data, I think. And I know you're working with Dan Belsky, who was just on the podcast not too long ago in the Biomarkers of Aging Consortium. Just, I mean, tell me a little bit about your work there and what you're thinking, and how, if there's any any
Okay.
any highlights on measuring biological age.
So two things. I think
Yeah.
so. We've measured 5,000 protein in 1,000 people in our studies.
Hmm.
Um, and basically, we published a lot about the proteomics that are increased between age 65 and 95. But the most exciting paper is a paper with Tony Weiss-Corey. It was in Nature last year.
Hmm.
And um, because when I looked at the data first, what was striking when we put them in pathways, there was breakdown. Okay, there is a breakdown of collagen and of platelets and of lots of stuff. And then because of the breakdown, we said, maybe what we have in our proteome in the blood is proteins that are coming from organs that are breaking down. So what are proteins that can only come from those organs? And we looked at 22 organs, and then we actually calculated not only the age, the biological age of the whole body, which we could do with those proteins, but actually the biological age of the organ. Now, if you're slow aging, basically all your organs will slow aging, and if you're fast aging, all of them will fast aging. Okay, but the correlation is 0.8. So I'm working now on a group of people who are slow agers, but their brain is fast ager. Okay, or liver ager. So why is your liver aging rapidly than that? Are you a drinker?
Right.
Do you have cancer?
Yeah.
And but I think the prospect of having not only your biological age, but also what organ might lead your aging, and can
Mm.
we focus on that, is
Yes.
going to be just terrific. Now,
Yes.
I would tell you that Dan Belsky and I just were invited to give a full report to ARPA-H for our projects, where in 18 months we are going to find all the biomarkers of aging. Now listen, that are changing with therapy. Okay, so we're going to get um, blood samples from DPP, from GLP-1, from SGLT2, from metformin, from all those drugs and intervention and lifestyle interventions. And we're going to see not only what is your biological age, but which have changed. Look, the epigenetic biomarkers are changing little or slow. Okay, probably proteomic and metabolomic will be much more important. So now every one of those therapy will do its own thing, but all of them together will have the biology of aging inside, and those will be turned in the major markers. So watch it for the next 18 months or 2 years. We'll have a major advance of how to measure aging that is clinically relevant, right? The whole idea is somebody is coming to you, you want in 3 months to show that they're going to the right direction.
Yeah. Yeah.
When we're developing when we're doing a phase 2 trial, we want to see that the biomarkers are going to the right direction before we waste money on phase 3.
And
So,
this is the fast, the
and this is
fast
the
trial.
past, right?
Awesome. Awesome. Well, you must be a fan of the pace of aging, his clock, the Dunedin pace of aging. I mean,
Absolutely. Absolutely. I think Dan Belsky is really is really the man to take us forward on this on this road. He's smart. He's a leader, and I'm glad that you had him in his program. I'm a fan.
yeah. Well, Dr. Barzilai, thank you a million times for joining me and going over and just covering all of this information in such a careful way. I think I certainly appreciate it. I just very much appreciate this careful exploration and really kind of, blowing up some of the misinformation that's being reported out there.
Good questions. I enjoyed it too, and best wishes. And
Thank you.
I hope it's helpful.
I want to personally invite you to the Functional Medicine is Longevity Medicine Masterclass, a free online event featuring the leading minds in functional and longevity medicine. We'll explore why functional medicine remains the most science-backed path to extending health span. In a world chasing the latest longevity hacks, we're spotlighting what we've known all along. Healthy longevity starts with root cause resolution, systems biology, and personalized care. Join us live September 17th through the 19th at functionalmedicineislongevitymedicine.com. Thanks to our Diamond Level sponsors. Precision Analytical, creator of the Dutch test, is offering evidence-based training on key topics like hormone metabolism, interpreting Dutch test results, and therapeutic strategies, including hormone replacement therapy, all aimed at improving patient outcomes. Become a registered provider to gain access to an expansive education hub with ongoing content, including webinars and case studies. Visit dutchtest.com/courses to get started and grow your practice. For over 40 years, the foundation of Biotics research has been innovation and quality. Their goals remain unchanged: innovative ideas, carefully researched concepts, and product development with advanced analytical and manufacturing techniques. Biotics nutritional products are of superior quality and effectiveness. They are available exclusively to the healthcare provider. Find them at bioticsresearch.com. Thanks to our gold level sponsors. Vibrant Wellness, transformative lab tests for wellness. Find them at vibrant-wellness.com. Timeline, fueling cellular energy for a lifetime of healthy aging. Find them at timeline.com. Disclaimer. This podcast is for informational purposes only. If you have a specific medical concern or if you have any questions, please speak with your healthcare provider.